Lecture 2, then we start to talk more about the technique, the recombinant DNA technology, but still some basic things here. Okay, so first of all, characteristic of genetic code. So again, what is gene expression is from DNA to protein, and with RNA as the intermediate, we have DNA replication, transcription, translation, a lot of enzymes involved. The final product is the protein, which is made according to the gene. Okay, so then that is the gene expression, and we have the two key steps here: transcription, translation, and if this is a eukaryote cell, RNA processing. So again, comparison, raw material, products, enzyme, where does it happen, something like that. Yeah. And then about genetic code. So genetic code is basically the ATCG, and then there are some characteristics of them, but we have covered it in the quiz, I guess. Yeah, so some keywords and then the explanation. Yeah, like they are within free, they are universal, they are found everywhere in every species, something like that. Then this is the characteristic of genetic code. I think we have talked about this in the quiz. And gene expression will lead to the protein synthesis. So I have explained that very clearly. And so this is still also, and this is a codon table. So you must know how to read it because you have learned it from year 1, I guess. Yeah. So how to read a codon table is that if I give you a sequence like that. So first of all, make sure it is a DNA sequence or RNA sequence, and this is the same as your codon table because we have DNA table, we have RNA table. So if they are the same, then they can be used directly. And then always find the starting one, unless if they ask you to start from the beginning, or if they tell you have to find the starting one, then you have to find the AUG, which is the start codon. Okay, so after you find the start codon, read it every 3 letters because genetic codon is triplets here, triplets. So you have to read it every 3 codon or every 3 letter, and then find the corresponding one from the codon table. Okay, if you forgot how to read it, you start from here, you find the first letter U, you start from U, and then you find the second letter A. Here, so here say first, second, and third. Okay, very clearly shown. So this one, UAU, you start from U here, and then A, so this box, and find the last letter here. Then you will know the amino acid to be added based on this codon. And then you move to the next 3. So don't overlap the 3 letters. So 3 letters, and then the next 3, and the next 3, until you reach a stop codon. Okay, so stop codon is always, so this is clearly shown here actually. Start codon, stop codon. So if you reach UAA, UAG, or UGA, then there will be a stop. So no amino acid will be added. You can just stop here, or you can write down a stop here. Okay, unless the question asks you to do everything, or else you have to find the start one and stop one. Write down all the amino acid in between for every 3 letters, and this is the codon table. Okay, so of course your body don't read a table, but they will follow the same code, and then they will make the amino acid sequence based on the DNA sequence, and then the amino acid will form a polypeptide. They are folded, they are processed, and then they become a functional protein at the end, hopefully. Okay, so by reading the table, so the amino acid can be linked together, they form polypeptide, and this is the process of the transcription. So transcription basically is to use the DNA template because they are too large to leave the nucleus, so they have to make a copy of a smaller, shorter strand of RNA, and at each time you don't have to copy the whole DNA to make a protein. You only need a small segment of them. So you make a copy by transcription, which is the copy is mRNA. If it is a prokaryote bacteria, then it is a messenger RNA. If it is a eukaryote like human, then this is a pmRNA, and there need to be further RNA processings for eukaryotes. And then next step, translation, and finally amino acid amino acid chain, polypeptide, and eventually protein. Okay, so transcription is basically making RNA from DNA, again, raw material, products, the 3 steps, which are the same as in transcription and translation. They are both called initiation, elongation, termination. So don't mess up with the PCR stage. Okay, PCR stage is also something like that, but that is denaturation, elongation, and annuling, and then elongation. So these are different 3 steps here. Okay, so the enzyme involved. The process, basically the process, the enzyme involved, phylopods. Okay, so these are like with some more detail, but actually I don't think you have to go that deep into like all the factor, TATA box. That's too deep for you. But make sure you know how it happened, the enzyme involved, the phylopods, something like that. Okay. Yeah, so a final determination how it is stopped. Yeah, the difference between prokaryote, eukaryotes for the process. Okay, and then some regulation, because the transcription won't happen all the time. So the cell needs some order to tell them when to start, when to stop, whether to make a lot of mRNA or take a break. So there are some mechanism to start or stop or speed up the whole thing. So some repressor, activator, so some basic idea on how it works. Okay, so basically if you have activator, it will like speed up the process. If you don't have the activator, it will not start. With repressor, it will stop. So just something like that. Like positive and negative control stuff. And then, so after you successfully make a mRNA, you may need to further process it in eukaryote. So you have to know what are those processes involved, adding something at both ends, splice it so that the unused part is removed, and then the coding part is joined back together. So the basic work, basic modification involved in the RNA processing and the function. But we also covered this already in the quiz. So why are they modified? Okay. And then, so after the RNA processing, you have a functional or mature mRNA now. And then the last step is translation. You make use of the mRNA and you make a polypeptide, which will further be modified, processed into a functional protein. So again, the 3 steps, and then some enzyme and some more RNA involved here. So the major one, the name of them, the ribosome, the tRNA, the template, and eventually the protein. Okay, so but these are like happen everywhere in many courses. So I think you are very familiar with the steps already. Okay, so that's the basic thing. Guess I draw one. Okay, and so talking about the ribosome, so the ribosome is this thing, this big thing. So there are large subunits, small subunits, and there is some interesting phenomenon happening is called polyribosome. It looks like this. Okay, so this is like they line up together, they are sharing the same mRNA with more than one ribosome sharing our mRNA. So this is an interesting picture you can see here. And so you have to know this is something called polyribosome. And what are they? Why are they important? So they can they speed up everything, okay? Because you don't have to wait for one ribosome to finish the step before the other one. So you can start many synthesis of protein in the same mRNA template with more than ribosome. So basically speeding up the whole process. And that's why we can make the protein fast enough like that. And for prokaryotic cell, it is further speed up because they don't have the nucleus, so the translation can start before transcription finish. But it is not possible in eukaryote because you have to allow the mRNA to leave the nucleus first. Okay, so these are some tricks, some tricks that they've speed it up in our body cell. Okay, so regulation prokaryote versus eukaryotes. Yeah, so that's everything here in lecture 2. And